I'm studying transcriptional regulators of metabolism relevant to diabetes and obesity and other aspects of metabolism. And our work focuses on a few different tissues, principally in the liver as well as in skeletal muscle. We have been studying this gene BCL6 for a number of years. Others had looked at the functions of BCL6 in the immune system where it's highly expressed. But one of the sites where it's most highly expressed is skeletal muscle, where its function has been entirely unknown. And so we decided to look at this. Usually when things are expressed at high levels, means that a gene is doing something important there. And so we used mouse genetic approaches to understand its function and found that it's very important in regulating skeletal muscle mass. We were able, using the power of mouse genetics, to specifically delete BCL6 in the skeletal muscle. And when we did that, we took two different approaches to doing that. We took an approach of deleting it in utero, in the developing mouse, and we took an approach of deleting it in the adult mouse. The first approach, which was deleting it in utero, we found that mice were somewhat smaller and their muscles were about 35% reduced in size than a normal mouse. And so we took this further and deleted this gene in the adult mouse and found the very same effect in skeletal muscle, that the muscles were reduced about a third in their mass. And with that, we found consistent effects on muscle strength. So not only was the size of the muscle reduced, but the strength was proportionately reduced. We took a number of approaches to understand how the sort of balance of muscle synthesis versus breakdown was affected by BCL6. We knew that when we lost this gene in muscle, that muscle quickly wasted away, but we didn't understand which part of this balance was affected. And one of the approaches that we took, which is a very powerful approach because it gives us definition about specific proteins that might be affected, was to use ribosomal sequencing, which is a sort of molecular genetic approach to look at what genes are being translated. And what we found is that when we lose BCL6, there is a very powerful impact on the translation of muscle proteins. So there's an important impact on muscle protein synthesis, affecting not every protein, but a large number. You know, our work on BCL6 really was the first to define this particular gene and placing it in the context of some other pathways that others had described as playing a role in this sort of balance of muscle protein synthesis and breakdown. It's only after we can really define the molecular players that are responsible for controlling this balance that we can kind of think about how can we tweak or target this system to hopefully improve outcomes in states that lead to muscle atrophy and loss.